Well, thanks. Yeah, thank you for inviting me and for being here. It's actually really nice for me to be back in Maastricht because, short anecdote, 10 years ago, I did my master's internship here. Very different type of story because I actually studied whether an intervention of cocaine made people more creative. Um, it did, but only in non-verbal ways because verbally they got worse. So yeah, that is really a different part of my history, let's say, because now I'm more into the gut-brain axis. And I figured since some of the people in the room, I've been citing their work for 10 years already, but I'm only meeting them face to face for the first time. And others of you I don't know at all, which is actually the nice thing about this smaller conference. We can get to know each other. So I figured I'll tell you a little bit of my sort of personal journey through the field of the gut-brain axis. Because when I started, I was actually a very young PhD student, and I just almost coincidentally started a study on probiotics and cognitive reactivity, and that sparked the rest of my research career after that. I now work as an assistant professor at Leiden University in the clinical psychology unit, which I guess also explains that today I'll focus mostly on mental health and like the gut-brain axis in mental health in different ways, I guess, than we've seen yesterday and this morning. And everything I'm telling today, almost everything, I guess, was funded by NWO, the Dutch Organization for Scientific Research. So why do I focus on mental health? Not only because I work in a clinical department, but also because the burden of mental disorders keeps increasing, right? I think we all, I won't not ask you to raise hands, but I think we all know someone who has suffered a depression, an anxiety disorder, maybe an eating disorder, even if it was perhaps yourself. And so the WHO already formulated mental health as one of the key points to improve back in 2019, and mental health is actually part of the Sustainable Development Goals now. So that's why we keep working on this. I guess most of us actually here. Yeah, where did it start? At least for me, or maybe this field. As far as I know, like the most recent sort of development, we've heard it this morning, the spark in the gut-brain axis research. I think it started more or less in 2004 when a group of researchers showed that germ-free mice who were exposed to stress showed a stronger stress response, a physiological stress response, than mice with normal microbiota. Germ-free mice, I think everyone knows, but they are mice that are kept sterile, so they basically don't have microbiota. And the intriguing thing was that when the same mice were given probiotics, the responsivity was reversed and it normalized. I didn't take the picture, but their responses started looking similar to healthy mice that were exposed to microbiota earlier. And we've heard the definition yesterday, but just a reminder for you, probiotics are live microorganisms that when you ingest them, at least have are intended to have health benefits when you consume them. So after this study in 2004, it took a while, but what you see in the literature is that there were many papers that started showing up saying that the gut-brain axis could be a key component in stress regulation. It was a missing link in depression. I think we talked about this study, or we saw this study this morning, that the gut microbiota, the gut-brain axis could be really the next big thing in mental health. And yeah, there are people that call it a paradigm shift in neuroscience. Now, the interesting thing that I found is that although this might be true, these papers were mostly theoretical or like perspective papers. And if there was any empirical work, it was only mice and rats. There was maybe 1, 2, 3, 4 human studies, but this was really only based on mice research or rodent research. So it was 2015, I guess, maybe a little bit before that, when I started looking into, okay, what is the evidence in humans? And if we have a link, we have an association, okay, that's nice. But is there also something as like a causal contribution or a direction? And what we saw after that were actually studies in both humans and mice that showed that, for example, when you transplant germ-free mice with the microbiota of depressed patients, they started showing depressive behavior. There were also some studies in humans, mostly depressed women, where they, for example, did an intervention with probiotics, and it showed that especially the women suffering depression with the really low moods, they improved when given probiotics, but not everyone improved. So there were some studies showing, okay, probiotics or the gut-brain axis could have some potential also in humans. And following those studies, especially the ones with interventions, we've also started to figure out what we saw this morning, namely how that could work, and that there are neural, endocrine, and immune signaling pathways between the gut and the brain, reciprocal. We also saw that this morning, that could not only explain but also help in how, or help understand how the gut and the gut microbiota could play a role in brain functioning and mental health. Now, the question, of course, and I don't recall who said it yesterday, if it was Nicole or Peter, but I recall someone said, when we're doing research or when we are focusing on probiotics, prebiotics, the focus should be on prevention. And I agree. So the question here still is, what is the preventive potential of the gut-brain axis? Could we use probiotics, prebiotics, postbiotics, short-chain fatty acids to perhaps prevent mental disorders? Now, if you want to dive into that question, where I start as a psychologist, as a clinical psychologist, is I look at the psychological mechanisms that we know predict, for example, the onset and development of depression. And one of the most established psychological mechanisms or factors that we know predicts how vulnerable people are to develop a depression is, for example, cognitive reactivity, and especially rumination. So cognitive reactivity, for those of you who are unfamiliar, is basically the degree to which you activate dysfunctional thought patterns when you are in a sad mood. And rumination is one of them. So for example, if you feel sad, are you the type of person that starts ruminating on, my life could have been so much different if only I hadn't done that? Why am I so sad? I should be feeling better. That factor, cognitive reactivity, really predicts how vulnerable you are to developing, but also maintaining depression if you're already a patient. So yeah, back in 2015, what I figured was, let's do a study with probiotics and see if probiotics can decrease this cognitive reactivity. We saw some studies in which probiotics could decrease depression symptoms. But at the time, and still actually, I was mostly working on prevention with healthy people, and prevention could potentially be in decreasing this cognitive reactivity to sad mood. So we just asked the question, can probiotics decrease cognitive reactivity to sad mood? And honestly, because we were one of the first to work with healthy humans, and there was mainly animal work and some patient work, it was kind of a wild study. It was really, in 2015, this was really considered controversial. I remember, so we actually found the effects, I'll show it. We found probiotics, 4 weeks of probiotics compared to 4 weeks of placebo to decrease cognitive reactivity to sad mood, and specifically aggressive thoughts and rumination thoughts. The nice thing is that after that, this study was replicated many times. It's always good as a researcher if it's replicated, but there's also a few studies showing that the effects can only be replicated or found in people who, for example, show high cognitive reactivity or show high responsiveness to stress. So the effect, certainly, as any effect, doesn't apply to everyone. Now, after 2015, like also Lucas just told, this was one of the studies that really fastly got cited so often, and I got invited to speak about it everywhere, even in conferences where probiotics were main topic. And the funny thing is, at least back in 2015, probiotics were main topic in livestock conferences. So I'd be presenting this study at a conference where people in the room were mainly working with developing probiotics for chickens or cows. It was really educational, to be honest, but yes, I was really considered, or this study was really considered controversial. And I think it's beautiful to see how over the past at least 10 years, we see that, well, more questions arose, but also we find more. There seems to be really something of the gut-brain axis in mental health. We don't know exactly what, but it's our job as scientists to study that, right? So at least it's not considered that controversial anymore. Otherwise, I wouldn't be standing here. I really recall if I would be in front of a room with my colleagues, with psychologists, the clinical psychologists. This is the group of people that go, oh, okay, have the brain. Okay, yes, that's involved. But then as soon as I would start talking about the gut, they'd be like, are you insane? Why are you talking about poop now? Because yes, that's also what we measure, right, in this field. Anyway, it just feels like a beautiful memory. So we found this effect. Probiotics, 4 weeks of probiotics compared to placebo decreased cognitive reactivity to sadness. I have to say that it was a very small sample. We had 20 people in every group. So 20 people taking probiotics for 4 weeks and 20 people taking a placebo for 4 weeks. Nevertheless, we found this and it was replicated also in larger samples. I'll get back to this study in a moment. How do we think, or what did we think at the time was happening? As a psychologist by training with a little background in cognitive neuroscience, I really had little knowledge on the gut, but I learned very quickly that the probiotics we used was a mixture of species. It had 9 different species of probiotics, which is what we call multi-species probiotic, and it was specifically designed, at least in vitro, to protect the gut barrier or the epithelium. So to basically keep toxic substances from leaking outside the gut into the systemic system, etc. So when I got into this picture, into this field, aside from assuming or hypothesizing perhaps that probiotics were indeed protecting the epithelium, which at that time I figured was another researchers group to further investigate. What we noticed was that, well, we noticed not necessarily, but what stands out is that the vagus nerve is a main component here. We've seen it quite often, I think almost in every talk yesterday and today, that the vagus nerve is a key component in this gut-brain communication both ways. However, still, when this picture was published, it shows the picture of a human brain, but it should have been a rat or a mouse brain, because also this system was basically, formulated based on mice and rat studies. We've heard it yesterday also from Aline that, cutting the vagus nerve, for example, to investigate the causal role in gut-to-brain communication and its role in mental health and effective functioning is basically impossible in humans. It is possible, though, because I was told that it happens sometimes accidentally when people get like a gastric bypass. Doctors sometimes accidentally cut the vagus nerve. Anyway, I've never continued on that research line, but what we did start to do is investigate the vagus nerve or the role of the vagus nerve in the various cognitive and affective components. And this was inspired by Porges, who phrased that the vagus nerve is a driving phylogenetic element in the ability to both monitor and regulate complex behaviors, but especially also attention and emotion. Now, this name is a bit controversial these days, actually, because I think many of you know the polyvagal theory, which is discussed and criticized. But nevertheless, we continued on. Back in the day, no one had heard of the polyvagal theory yet, actually. And we continued, or we started to investigate, the role of vagal nerve stimulation in various cognitive and affective mechanisms, or let's say functions, using transcutaneous vagal nerve stimulation, which I think you had a very elaborate explanation on yesterday. But basically, we can apply an electrode to the ear, the left ear, to stimulate efferent fibers of the vagus nerve, the vagal nerve. And because it is supposed to be a driving element in attentional, cognitive, and emotional processes, we looked into basically questions of which parts of that, or are there functions that the vagus nerve stimulating the vagus nerve can stimulate or can increase. And I brought to you some of the findings. For example, one of the things we found was that when people, these studies are, by the way, always done using a placebo-controlled design. So people come to our lab twice, and once they get a placebo or a sham stimulation, basically what we do is we flip the electrode and we stimulate the earlobe. You have to be careful with that because it cannot touch the cheek, then you're stimulating something else. But the earlobe is relatively, or it's not relatively, is out of efferent fibers of the vagal nerve. So people come to the lab twice, once with a sham or placebo stimulation, and the second time, or the other way around, of course, they get active stimulation. There are some exceptions because some tasks cannot really be done twice because people are always, but in general, we always try to do it this way. And one of the things we found was that when we applied vagal nerve stimulation to people, the degree or the extent to which they started following other people's gazes increased. So we always follow other people's gazes because it could tell us something important. If you're all looking at me and I suddenly look that way, even if I don't turn my head but only my face, that is information to you. There might be something dangerous or something important there. And stimulating the vagal nerve increased the amount, or let's say the number of times that people follow that gaze. We also did a manipulation to see if the emotion that the person who was changing their gaze was expressing was doing something, but nothing happened there. So regardless of the emotion that people were expressing, the people who received active TVNS followed the gaze of other people more. So this says something about, let's say, the deployment of social attention. We also found in various studies, not only we, also others, that TVNS can increase people's ability to recognize emotions, mostly in bodies, to be honest, less so in faces, but more in bodies. So this is both static bodies, but also moving bodies. There's one exception I found in my own study, that when we apply active TVNS, people become worse in recognizing sadness in bodies. That might make some sense from an evolutionary perspective, but it's too much to go into that now. But that means that stimulating the vagus nerve doesn't simply improve your ability to recognize everything. There are subtle manipulations and differences between emotions, for example. I find this a really interesting study from another lab, from Crystal Fong's lab. And she showed that active stimulation with TVNS decreases the intensity of the emotion that people report when they are demonstrated a scary or an emotional stimulus, for example, a snake. So it decreases the intensity that people report because they ask people to reappraise. And the TVNS kind of stimulates or supports the cognitive reappraisal of emotions. And they think thereby lowers the intensity because people reported lower intensity of emotions they were feeling. The interesting thing about this is that the physiological response to the emotion or to the picture did not change. So heart rate and skin responses were not changed under the influence of TVNS, only the subjective intensity that people reported. I just want to keep that in mind because I'll get back to that in a moment. Just for your information, this ability to recognize faces better, we recently also demonstrated that after probiotic intervention. So, of course, the next question is there some overlap in what maybe stimulating the vagus nerve may do versus what probiotics or prebiotics or any positive intervention in the gut might do? I inserted this slide late yesterday because yesterday Aline showed us that probiotics could manipulate or could at least affect the degree to which people are willing to take a risk. And especially if I recall you correctly, probiotics might decrease or might enable people more to regulate the emotional responses that come with risk taking. And that reminded me of another TVNS study we did, namely that active stimulation of the vagus nerve affected delayed discounting. And delayed discounting is basically your ability to delay rewards. It's a form of non-risk taking, actually, or more risk taking, depends on how you look at it. And we specifically found that was moderated by your current mood. And I'm not sure if I can already explain completely how this might work, but thinking of your finding yesterday, I suddenly figured there might be a relation here because we're both still talking about gut-brain axis and finding something on risk-taking and emotional regulation of that. So I think we should talk and maybe set up a future study. Going further, I had this TVNS, which was mainly research I was doing during my PhD. And then after that, well, first of all, COVID hit. And also, to be really honest, TVNS studies are a little bit easier for me to conduct than probiotic studies because they are quicker. It's acute stimulation of the vagus nerve. Probiotic studies are often like at least 4 weeks. Recruitment is harder, etc., etc. But nevertheless, I think it was like 2024 or something where I figured, you know what, let's go back to the beginning. What did I really learn? Not just what did we learn as a field, because I've been in many of these meetings. Where's the field right now? Where should we go? What have we learned? But I just started sort of self-reflection. What have I learned and what should we do? And then I figured maybe I should go back to the beginning and just see if I can replicate my first study. Because I saw other labs doing it, sometimes showing slightly different effects, for example, not on rumination, but on risk-taking or on control thoughts, more controlling thoughts. So yeah, I replicated that study. What we did is we had, again, exactly the same, a 28-day probiotic intervention, 1 week, one group of people with a pre and post assessment taking a placebo, and the same group, but then taking a probiotic. Same probiotic mixture, just more tasks, because in the meantime we had learned more and we wanted to figure out, for example, if probiotics could affect the ability to recognize faces. We measured that using the facial expression recognition task from Catherine Harmer's group. And we measured some extra questionnaires and also emotional cognitive tasks. But these measures, the state trait anxiety index, the LIGHTS-R is a measure of cognitive reactivity, and the CSD were similar measures to the first study. I think every one of you recognizes the Maya. It was actually the Maya too, because we also wanted to know whether probiotics would affect interoceptive awareness, at least, or perhaps alexithymia. And also aggression, because in our first study we found something on aggressive thoughts. So now I wanted to figure out if it also perhaps changes aggressive behavior. These are basically our sample characteristics. We had double the sample size, so 44 in every group. No differences when looking at gender distribution and age and stuff. The surprising part here, when we go to our findings, is that we found exactly nothing. The light lines is the placebo, the darker lines is the probiotic. If anything, we found that the placebo group started worrying a little bit less in the post-intervention assessment. So yeah, the placebo improved in the sense that they worried less, but probiotics did exactly nothing. So then, yeah, you scratch behind your ears and you're like, what's happening here? We have the Maya and the alexithymia and the aggression questionnaires here. We again found nothing except for the probiotic group decreasing their not distracting interoceptive awareness scores a little bit. But that was really the only thing we found. And if you look at the number of questionnaires we took, this could as well just be a multiple comparisons. It would not survive that correction, let's put it like that. So yeah, we found nothing. And then what? Now, if you paid attention to this slide here, you might have noticed that we also took daily assessments. So where we didn't do that the first time, this time I wanted to know at the very least, does something change in their, their stool, for example, the frequency of their stool, or maybe the the consistency, so how much water their stool contains, or do they have like some bowel complaints? And I also wanted to know for sure. We told them it's okay if you don't take a dose, but just write it down. Which funnily enough made almost everyone finish all the doses. And they also brought back like the little box with the sackets, and there were, yeah. But we also, I thought, we're asking them 2-3 questions every day regarding their stool. Literally, what did your poop look like today? Did you go to the toilet or not? I figured let's also ask them, how did you feel today? Very crude measure. We just asked how many positive feelings did you have today and how many negative feelings did you have today. It is super crude, but yeah, we had to do something. And then my postdoc, Katerina, after this sort of disappointment, she sat down and looked at the daily measures, and she found a decrease there. So we asked for positive feelings and negative feelings. For the positive feelings, nothing happens. There's no group or time interaction here. But for the negative feelings, we find a significant group by time interaction, meaning that if you look at this, the blue one is the probiotics. The probiotics group after 15 days basically starts showing a decrease in negative feelings. At first, we were like, it was a very crude measure, but at the same time, we cannot deny that we measure this effect, or at least the trend, basically for the past 13 days. So it's not a one-time measurement, not even a 2-time measurement. It's a 13-time measurement from day 15 to 28. We see basically nothing happening from 0 to 15. So yeah, that was our observed effect. And then what? What does it mean? Before we started further interpreting that or discussing that, we also looked at what predicts, how much people improve or how much people decrease in negative feelings in this case. By the way, what you also see here is that there's no, positive and negative feelings are not on the same scale, right? We see no differences or no changes occurring in positive feelings, but we do see something happening in the negative feelings. and they're not necessarily correlated to each other. So what we found was that the change in the probiotic group in terms of negative feelings, so how much people decreased over time in reporting negative feelings, was predicted by their baseline Maya not distracting score and the lights are risk aversion score at baseline. Now, when we looked at regression models, only the risk aversion score came out as a significant predictor, meaning that those people in the probiotic group that had a higher risk aversion score showed a stronger decrease in negative feelings during the probiotic intervention. And now I'm kind of going back also to yesterday's talk and yesterday's findings, because what is this risk aversion about? This is about how you avoid emotional difficulties, and especially when you are already in a somewhat sad mood. So, for example, statements from that questionnaire are, when I feel down, I'm more busy trying to keep images and thoughts at bay. Or I feel more like escaping everything, or I'm more inclined to avoid difficulties or conflicts. So this is basically a person's tendency to avoid further difficult emotions and risks, and especially emotional risks. So therefore, I figured yesterday, indeed, maybe probiotics may help regulate emotional signals that are related to uncertain outcomes, like you also reported. And this would also make sense in terms of a novel stress theory that has been introduced in 2017. I don't know if it's familiar to you, but it's the generalized unsafety theory of stress, which states that the stress response is not something that's activated when we're presented with a stressor, but it's actually the reverse, where the stress response is usually inhibited and it's disinhibited whenever we don't feel safe. And this might somehow relate to the effect that probiotics and the gut-brain axis have on stress. But of course, we need to definitely dig further in order to understand what's happening here. So where did we take this? We found this effect on a decrease in negative feelings following a probiotic intervention during a probiotic intervention. But that stands in a very sharp contrast with finding no effects when comparing pre to post intervention assessments of anxiety, even the PANOS, the positive and negative effect scale. So we went back to what actually did we ask people. So first of all, we asked them about their feelings and not necessarily their mood. And even for me, I think some of you might naturally be like, yes, there's a huge difference there. Where mood is somewhat more stable or more subtle, perhaps, and feelings are, I don't know, maybe more physiological. I don't know, but we specifically asked about feelings and not mood. Whereas a lot of measurements we do are usually about mood. The other thing is that despite the temporal, let's say we measured it more often, so we know for sure that there's 15 measurements and not just one coincidental finding of this. Although that is a strong side of this question, the other thing is that we have no idea what negative feelings means to people. So for some people, the answer to this question might have been about anxiety. For other people, it might have been about being tired. For yet other people, it might have been about being sad. And that is what we do in standardized measures, right? We rip everything apart and we ask them, are you sad? Are you tired? Are you anxious? Are you nervous? And then we find nothing. But here we basically put everything in one big heap and we tell them, did you have negative feelings? And maybe the strength of this question is especially in the non-distinction between was it tired, was it sad, was it anxious, etc. Now, scientifically, that's really tricky because we don't really know what this means. However, personally, and when we talk about personalized approaches, this is what is most meaningful to people. Quite often, people also don't know whether the bad feeling they have is because they're tired or because they're sad or anxious or maybe everything mixed. So from a personal perspective, this does sound meaningful. And that kind of brings me to the last point that I want to make, which is a little bit different, I guess, than from what we heard before, but nevertheless complementary, is that if we want to know, or if we ask the question, what is the potential of the gut-brain axis for mental health, then we have to realize that mental health is not only about behavioral and or physiological responses, things we see and measure on the outside, but it's also really about mental experiences people have. Those of you working with patients can also tell that sometimes patients objectively don't do any better, but they suddenly report that they feel better, or the other way around. They should be doing better and they might be behaving better, but they don't feel any better. So people have feelings about their feelings. There is something to this subjective mental experience that might actually be, interfering with what we measure and observe sometimes. And the tricky thing is that when we are thinking about psychological or psychiatric interventions, for example, asking whether a probiotic could ever be a replacement for an antidepressant, I don't even think we should, consider that as a question. Again, focuses on prevention. But when we ask that question, it is about subjective mental experiences. Because when we look, for example, at the effect of antidepressives, it's about whether people actually feel better. We should be asking the same question, I guess, when it comes to prebiotics and probiotics or interventions of the gut brain axis, at least in addition to the physiological and behavioral measures we're doing. So basically, we have to put the mental back in mental health. And that's really tricky scientifically, because how do you allow a very subjective experience or feeling, a feeling about a feeling or a thought about a feeling, how can that be measured and taken into account in the measures we're taking? That's objectively really tricky. But nevertheless, if we want to ask this question, if we want to get to really personalized approaches, we will have to, because a really personalized approach means that the person is allowed to have a feeling or a thought about what we are putting onto them and what we are making them do. So I think that this is the next big challenge, not just for this field, but for the whole field of psychiatric research, I guess, we're doing. Yeah, that's it, I guess. Yeah, we have this mechanism so far, and I guess the only last thing I want to put there is that this is not only a brain that's there, but there's a person around that brain, right? So it's not only brain responses and behavioral responses and physiology, but there's a person feeling something about those things. And what we shouldn't forget after all those measurements is to ask that person, how do you feel? Because we actually often do. Even I did that. I will now never forget to ask people how they're feeling during my experiments anymore. And just a nice quote to, I guess, send you home with, because Jill Taylor, one of the most famous neuroscientists of our time, once said that most of us think of ourselves as thinking creatures that feel, but actually we're feeling creatures that think. So keep that in mind when designing your next experiment, I guess. Thank you. I didn't notice who was first.
Thank you, but you were perfectly on time.
Yeah.
So, let's take some questions. Maybe
Yes,
that's
Okay, thank you, Laura, for a very nice talk. I mean, as a psychiatrist turned into a mechanistic researcher, I obviously agree with what you're saying about the importance of asking people how they feel and self-report. I mean, I think Mike can testify that every time in a biomedical context, people are like, yeah, but this is subjective and this is hard to measure. I mean, it's important. In mental health, it's probably the most important thing. So we should indeed include those things in our studies. That being said, I think most of us do. I don't think it's overlooked or overshadowed necessarily by well, maybe sometimes by physiological measurements, but I mean, in any case, I think it's important. I have two questions though. The first one is about every time I see people present about the transauricular vagal stimulation, I ask the same question and I never really had a satisfying answer to it. What is that branch doing there? And I mean, physiologically, what is it doing? And is this actually a good model for the gut brain? I mean, this is a totally different branch. Then I mean, it's a totally different. I understand it's convenient and it's accessible. But what does that tell us about vagal signaling from the gut?
The least satisfying answer, I guess, is I also don't know. But yesterday when we had to talk about the gut-brain coupling, no, not the gut, the stomach-brain coupling, I guess we also figured out we have to do a study to see if vagal active vagal stimulation changes that or does something there, would be the next question. Anyway, your first question, what is it doing there? I'm not an expert in this, but I do know that in cats, they actually and it relates to a more evolutionary perspective, they found that cats are really able to, first of all, they have this extra sort of ear part. I don't know if you've ever seen it or heard of it. And the idea is that they can move their ears, that part of their ears, to basically detect or to use it to hear if there's not only hear but also sense if there's danger. In that perspective, it would make sense that there is a branch there, and especially if it can be related to fight-flight behavior. Why we humans then have it, I don't know. I'm really curious also. Sounds unrelated, but some humans can actually move their ears. If that might be related to that same maybe rudimental kind of, I don't know, reason why that branch is there. But I don't think there's anyone who has ever really explicitly asked or answered that question, why is it there and what is it doing there? So again, I'm sorry, it's unsatisfactory, but there
Yeah, I
are
guess
some
what
ideas.
could be done, I mean, definitely there's also, when it comes to vagal and interoceptive pathway, there's some degree of somatotopy in the
Yeah.
posterior insular cortex and stuff. So I guess that's going somewhere else than where the gut signals
Yeah.
are going ultimately. At least, yeah, with people with like implanted vagal nerve stimulators for some reason, you could maybe try to compare stimulation of the ear branch with the implanted stimulator and then look at the brain. I mean, there's maybe ways to look into this, but yeah.
And in that sense, there have been fMRI studies where they show that stimulation especially activates, I think, the locus coeruleus, and like more afferent related networks.
And then upwards probably.
But
so.
And then there's, I mean, I'm talking about one way of stimulation, but there's different ways, right? You can also see that we use like the because there's also the inner ear electrodes. And then I guess another question is also like, if you do want to follow sort of a polyvagal perspective, then which of the fibers are really stimulated? We don't know.
And a bit related to that, last part of the question is how much is blinding an issue here, because you do have a control condition, which is nice, but people do feel the difference, I guess.
So we actually do take measurements to ask them to guess which session is sham and which is active, and they're usually around 50%.
Okay, so it's 52%,
but it's not significant. No, that is actually, and it also as a researcher, it really feels like an acting, like an act sometimes. And it also means that we have to exclude always any participant that has already participated in these studies before. So it makes finding new participants trickier. But we do check it.
But you have data showing that they're not better than chance.
We report it in the papers as well, because obviously.
And you could even try to, I mean, explore whether in people who did guess correctly, whether you see a different pattern from the people who don't guess correctly, I guess.
Yeah, good point. Because there's half of the people guessing correctly. That's true.
I actually like to follow up, and maybe it's a question for both of you. I mean, I'm super intrigued about the fact that you conclude about subjective experiences. Because I also believe that this is thoroughly missed in many evaluations. But the problem is, the minute you ask these questions to people, you make it an explicit or a conscious.
Yes, it is an intervention in a way.
And so I'm trying to figure out what we can do to actually monitor this while they're doing a task. And have you both considered to do EMA measurements or do some kind of brain state analysis, looking into shifts, these types of things that could be objective in a sense, but mirroring what we then ask from participants about their subjective experience.
Yeah, I would love to do a study like that. I mean, here we don't find the same effect in the placebo group, but I do agree with you that something happens the moment you ask people. I also think, honestly, that compared to other studies, we had a very, let's say, only 2 people dropped out of the study. It's a 4-week intervention. That's a lot. I think it's because we contacted them every day. So we asked them every day on an app to fill out, hey, how are you doing? Can you fill out like that? I think it does something. It is an intervention in a way. But how would you, I don't know how to objectively,
Maybe, I mean, we are using EMA in quite a few studies where, I mean, that's still different from what you guys are doing. Like people, not for 4 weeks in a row, but because that doesn't work, but like typically 1 week or so where people get random beeps at random times and they have to answer a few quick questions. So we are running a big, Annalena, the PhD student whom I showed, is running a big study longitudinal study where we measure them 3 times with 3 months in between. They do 1 week and we get microbiota data. So that's exactly aiming to look at how microbiota composition and function and a few mediators may be related to everyday subjective experiences of stress and emotions and things like that. So,
but what would you think about maybe using thermal measures, for example? So skin conductance or really body temperature, something that is potentially more reflective of
we are also including like wearables in many of these studies where we do measure some ambulatory physiological signals. Skin conductance, like very long measurements of skin conductance, I'm not sure how useful that is. But we do have heart rate and temperature and a number of these things with the idea that you have some ambulatory physiological measures, you have the self-report data, and you can look at both or the interaction.
Yeah, we actually have a really large EMA study in Leiden, the WARN-D study. I don't know if you've ever heard of it, but we have about the WARN-D study, early warning system for depression study. And there we followed, I think, about 3,000 people for 4 months with EMA like prompts 4 times a day. We haven't asked this question, but we could look at that because we also have heart rate data. But it's messy when people wear that, but still, yeah. Now, actually related to this study, it's something that I was struggling with, and this study wasn't the right one to dive into that, but I've been wondering whether just the intervention of asking people every day, how are you feeling, whether that perhaps interacted with the probiotic effect in a way that we indeed don't see any differences pre and post assessment. Because just as a thought experiment, if you think about like becoming better at emotion regulation, that might just result in nothing. Because you see, so I would really, I guess we have to figure out how we could measure that, but that might be interesting.
There's never gonna be something that has a one-on-one relationship with how you feel. And I agree that how you feel is what determines whether you go to work, whether you go to the doctor, whether you take medicine or not. So I agree that it's kind of the most important thing, but I don't think there's a way around it.
Well, I mean, one thing that we're currently interpreting,
we have
just ever so briefly, but to use into brain state, resting state dynamics, because you see very nicely transitions and durations and things like that. So if one can link that to all the subjective data that you measure from the participants on a daily. I mean, in a sense, you would have to include it with wearables.
The DMN comes out quite familiar, but anyway.